Hi everyone, and welcome to our video on some exam questions related to cell biology. Our first exam question states, the diagram shows a method used to grow pure cultures of a bacterium, and we're shown the method here. Okay, now it says name apparatus A and apparatus B. So as you can see, apparatus A is over here and apparatus B is over here. Now apparatus A is a loop. It's called the inoculating loop, and that's what you need to write down here. So it's called the inoculating loop. And apparatus B is the petri dish itself, which you sterilize cultures in. So apparatus B is the petri dish. And that would get you 2 marks. Okay, now the next question says, why should apparatus A and B be sterilized before they are used? So we're using them in the practical. They need to be sterilized to remove any harmful microorganisms. Okay, we don't want anything to affect our results, so it's to remove any harmful microorganisms. Okay, and that would get you 1 mark. The next question says, how should apparatus A be sterilized? Now apparatus A was the inoculating loop. So just as a reminder, it's the inoculating loop. How should it be sterilized? Well, the inoculating loop must be sterilized using a flame. It should be passed under a flame to remove any harmful microorganisms. That would get you 1 mark. Now it says adhesive tape is used to secure the lid on apparatus B. So remember, apparatus B is the petri dish, and it says give 1 reason why the lid of apparatus B should be securely taped. So again, we are growing bacteria, we are culturing it in the petri dish. We don't want any of that to escape. So we don't want any harmful pathogens from the petri dish to escape. If they escape, they can cause a real issue in the surroundings. So 1 reason is to prevent harmful pathogens from escaping. Okay, and that would get you 1 mark. Now the final question for this question is, what is the maximum temperature that should be used in schools to grow the bacteria in apparatus B? So remember, apparatus B is the petri dish. So in a school, it should be grown at 25 degrees. And this is to prevent too much bacteria and preventing the risk of any harmful pathogens being released. Okay, and that would get you 1 mark. Okay, next question. It says human cells and yeast cells have some parts that are the same. The diagram shows a yeast cell. Parts A and B are found in human cells and in yeast cells. On the diagram, label parts A and B. So parts A, as you can see, is the nucleus, and part B is just inside, and you can see that it's the cell membrane. And that would get you 2 marks. Now it says many types of cell can divide to form new cells. Some cells in human skin can divide to make new skin cells, why do human skin cells need to divide? So remember, they need to divide for growth and repair of body cells. Okay, so they divide by mitosis for growth and repair of body cells, and that would get you 1 mark. The next question says human stem cells can develop into many types of different human cells. Use the correct answer from the box to complete the sentence. Human stem cells may come from what? So we have embryos, hair, or nerve cells. So the most obvious answer there is going to be embryos. Human stem cells come from embryos for 1 mark. They can't come from hair and they can't come from nerve cells, as that is one of the specialized cells in our body. Final part to this question, use the correct answer from the box to complete the sentence. Human stem cells can be used to treat what? They can be used to treat paralysis. Okay, so you look at cystic fibrosis and polydactyly in inheritance. There's something which are genetically inherited. Okay, and that would get you 1 mark. Next question, it says the image below shows some muscle cells from the wall of the stomach as seen through a light microscope. Describe the function of muscle cells in the wall of the stomach. So what do muscle cells do? Well, muscle cells can shorten or contract. That's an obvious use. They can shorten or contract. And what else? It's saying describe the function in the stomach. So why does the stomach need muscle cells? It's to churn up the food which comes in. Okay, so the muscle cells, they churn up the food coming into the stomach. Okay, and that would get you 2 marks. Okay, so it says the figure above is highly magnified. The scale bar represents 0.1 millimeters. Use a ruler to measure the length of the scale bar and then calculate the magnification of the figure above. So this figure, we need to calculate the magnification. Now this is the scale bar over here. It's saying that this distance is 0.1 millimeters. If we measured that with a ruler, we would find that the actual distance is 40 millimeters. So the actual distance of this scale bar is 40 millimeters, but on the image size of it is shown as 0.1 millimeters. Okay, we need to calculate the magnification of this image. So how many cells are there? There's 1, 2, 3, 4. So there are 4 cells, and we are given the scale bar of 0.1 millimeters, which is actually 40 millimeters. Okay, so we know that each of these cells are 0.1 millimeters long, and we know that there are 4 cells. So the total image size is 0.4 millimeters according to this scale over here. Okay, and you need to use a calculator. So if the image size is 0.1 multiplied by 4, which is 0.4 millimeters, what would the actual size be? So it's 40 millimeters, the actual size of this, and there are 4 cells. So 40 multiplied by 4 gives you 160. Now we need to work out the divisibility over here, because it's saying work out the magnification. So all you would do is 160, what we've worked out the actual size as, divided by 0.4. Okay, so over here we would do 160 divided by 0.4, and that would get us an answer of 400. So the magnification is 400 times. The image has been magnified 400 times. Okay, next question. It says the muscle cells in the figure above contain many mitochondria. What is the function of mitochondria? And it's worth 2 marks. So mitochondria, the function is to release energy. That would be 1 mark. To release energy in what? It is to release energy during aerobic respiration. So your 2 marks are coming to release energy and aerobic respiration. Okay, final questions now. The muscle cells also contain ribosomes. The ribosomes cannot be seen in the figure above. What is the function of a ribosome? So ribosomes are present to make proteins. So they are there to make the proteins for the cell. You could have also put something like protein synthesis. And that would get you 1 mark. Final question. Suggest why the ribosomes cannot be seen through a light microscope. It's because they are really small. You need an electron microscope with a higher magnification and resolution to see ribosomes. So ribosomes cannot be seen through a light microscope because they are too small. And that would get you 1 mark. And that is it for this video. Thanks for watching. I hope you liked it. And one last thing, please subscribe, hit the like button and the notification bell.